elite cultivar level yields. So how I'm going to do this? Well, I have my association mapping panel, and it has 300 advanced lines from the barley breeding programs of Oregon State University and the University of Minnesota. They're all getting genotype with the 9K. They are all winter or facultative, mostly facultative 6-row barley. So for those of, yeah, Pat talked about the 2-row versus 6-row, and winter and facultative too. So I guess I don't have to explain that, but they have been selected for disease resistance, malting, and yield, of course. So the experiments that I'm running, here's a summary of the trials. Actually, there's been a change. It's 2 years for disease trials, not 1 now. But so there's a trial for each, or there's an experiment for each trait that I'm doing. And then notice, of course, for the nitrogen use efficiency and the water use efficiency, there's 2 treatments. There's the high, or really just the standard recommended treatment, and then a reduced. So for nitrogen, 70% of recommended, and water use, 60% of recommended. And we went and talked to an irrigation specialist in engineering, and he actually said that, yeah, 50 to 60% was a good range for water stress without actually getting into drought stress. We should get good results with that. And luckily, it was the easiest to set up for irrigation schemes. But and so then, of course, what we're going to start non-focusing to determine nitrogen use efficiency is, the yields that the lines are getting at these reduced treatments. How does that compare to the yields that they're getting at the standard treatments? And hopefully, is it the same for some of them, and not for others, because we don't want traits to be fixed. But so then, of course, there's now I have these CSR explanation slides, and I see most of you actually are working on CSR or working with someone who works on CSR, but bear with this very simplified explanation of it. So what's CSR? CSR is canopy spectral reflectance. And what is that? Well, next page. So that is, of course, for any wavelength in a given spectrum of the incoming solar radiation, what percent of that is being reflected back. And there are certain, and we measure reflectance, but we're kind of more, what we're looking at are the wavelengths at which, depending on the nitrogen status for NUE, depending on the nitrogen status of the plant, is the plant absorbing more at those wavelengths? If it has a higher nitrogen status, and it, of course, absorbance is a property of reflectance. And then we're comparing those to the kind of normalizer constant wavelengths in the near infrared range, and expressing again, this is oversimplification. I just gave this for a departmental seminar. That the data is essentially indices that are essentially a ratio of the nitrogen status, reflection at the nitrogen status indicator wavelengths compared to normalizer wavelengths. And then similarly for water use efficiency, there is the spectrum range at which water absorbs light, and comparing the reflectance at those wavelengths versus the normalizer wavelengths. So a little bit of the results from the summer. I'm still doing some of the field effect adjustments, but these have been adjusted already. So to just looking at for the nitrogen use efficiency trials, of course, for nitrogen use efficiency, again, I'm looking at the ability of, or genes that confer the ability to reduce, or alleles that confer the ability to reduce the input levels without reducing yield or yield quality. And so then for yield, I'd be looking at the difference between the yield at the low nitrogen level minus the yield at the high nitrogen level. And of course, the more negative this difference is, the worse the nitrogen use efficiency of the line is. the less negative this difference is, the better the nitrogen use efficiency of the line. And so you can see in the histogram here, there's a nice spread of, nice normal looking spread of differences between the lines, and these are our checks lying in the middle. And then similarly for protein, which is also grain protein, which is also strongly affected by nitrogen levels. And then there's the difference between the protein at the low nitrogen level minus the protein at the high nitrogen level. And of course, the more negative this value is, the worse the nitrogen use efficiency is. Higher the value is, the better the nitrogen use efficiency is. And again, there's a nice spread of phenotypic values, which hopefully will be due mostly to genotype, and can map some alleles for nitrogen use efficiency. And just hot off the press this morning, I don't have heritability values for the differences specifically yet, but my current heritability estimates for yields due to genotype, or yeah, for just yield overall, it's 0.45, which and then for protein, 0.24, and for heading heritability is of course quite high. It's 0.98, and for height it was 0.72. So that gives you an idea of the variation, or hopefully we control the variation relatively well in this experiment. And these are all elite lines, so they wouldn't be expected to necessarily vary wildly across, or widely across the lines. So that is it for me, and I will hand it over to Ryan.
Okay. Hello, everybody. My name is Ryan Grabner, and today I'm going to talk to you about the genetics of beer flavor. Now, just so I'm sure you're all very familiar with the beer making process. Oh, I need to get on to the next. There we go. I'm sure that many of you are very familiar with the beer making process, but just so that we're on the same page, I'm going to go over how to make beer very quickly. First, you've got let's see. First, you've got the grower up here that produces the barley. The barley is then sent to a malt house, and it is later where the grain is allowed to germinate and is later dried out. And then finally, it is sent to a brewery where the bulk of the beer making process takes place. So, in general, most stages of the beer making process have been very well researched. However, one area that we feel has been neglected is back up here with barley genetics. So in the past, breeding barley for beer has been all about improving malt quality and eliminating poor flavors that can pass from barley onto beer. However, our concern is that in this process we have missed or even selected against alleles in barley that could instead contribute positive flavors to beer. To get at the question of whether barley can contribute positive flavors to beer in the first place, we turn to land races and cultivars in the Barley World Core collection. We decided to use this collection for two reasons. First, as I mentioned before, we're concerned that alleles that could give positive flavors to beer have been lost for modern malting varieties. And if this is true, then the fact that these lines have had little or no modern breeding will work, or will be very much in our favor. And second, these accessions are very diverse. And here we've got a PCA plot showing accessions from the World Core, where the blue points represent the accessions we selected from our project, and the gray points represent all the other accessions in the collection. And as you can see here, we've got a nice mix of 2 rows, 6 rows. The little circle in the middle appears to be high altitude varieties as far as I can tell, and Asian food varieties. And a nice mix of some other stuff in between. The one area that we weren't able to sample very well was the Asian food varieties, and that was because the lines did not yield enough grain for our purposes when they were grown in the Willamette Valley. In addition to genetic diversity, we were able to maintain a large degree of geographic diversity in our selections. As you can see here, we've got Europe pretty well covered, as well as a nice spread from the rest of the world. As for our methods, this year the World Core was grown here as part of the TCAP, and at the end of the growing season, we harvested the accessions that produced a reasonable amount of grain for our analysis. From these lines, we selected 114 genetically diverse accessions that were recently sent to Minnesota to be malted. Once we've malted the accessions, we'll use a combination of machines and human taste panels to assess both the chemistry and the taste characteristics of these lines. And next spring, we plan to replant the most interesting accessions in 3 locations. And this will give us both more grain for our analyses, and then it will give us a better understanding of G by E, which I think will be crucial in validating our results. So, where is this going? Right now, our goal is to assess if barley genetics can indeed make positive contributions to beer flavor. If we confirm this in the future, we hope to continue our project, but with a much higher replication rate. And this will allow us to carry out genome-wide association scanning for alleles that can positively affect beer flavor. Then the next step, introgress these alleles into malting varieties and get better beer. And with that, we're on to Alfonso.
Hello, can you hear me? My name is Alfonso Cuesta Marcos, and I will explain briefly how we are making this transition from 6-row to 2-row in our malt quality breeding program. As Pat said, we need a rapid response and also an efficient response. And to accomplish those 2 goals, we are trying to implement a double haploid genomic selection program. With the genomic selection, we want to achieve good efficiency. We hope to achieve it. And with the double haploids, we want to advance generations very fast. We have started our double haploid genomic selection process last year. We are now developing the training populations, and we will start with the same process our food quality program next year. So genomic selection is a method that we use to estimate breeding values for individuals based on many markers distributed across the genome. And the breeding values of all those individuals are derived from marker effects that are estimated from a training population for which both marker and phenotypic data exist. The primary benefits of genomic selections are that selection can be imposed for quantitative traits very early in the breeding process, and thus substantially reducing the breeding cycle time. If we can couple our genomic selection program with the production of double haploids, we can even reduce this breeding cycle to a single year, because double haploids you achieve homozygosity, complete homozygosity in a single year. So every double haploid line is potentially a variety. This is the scheme of our double haploid genomic selection program. We have our training population, for which we will get genotype and phenotype information, and from this training population we will develop a prediction model. We will develop a prediction model and use cross-validation Sorry, can you hear me? Ah, thank you. Sorry, how about the pointer? Yeah. So before applying the prediction model to our breeding population, we need to uh, validate the prediction model. We are doing this now using cross-validation. So the double haploids that we will produce in the future and that we are producing now, using only genotypic information, we can predict the breeding value. So for the next generation, we will select in the double haploids with the best breeding value, and also we will select which double haploids will be intermated for the next cycle of genomic selection. Oh, there's something here. Okay, so this is an example of how the genomic selection works. We have in our training population, we have estimated the effect of all the markers that we have in the population. So we have estimated the effect of each individual marker. Then we have a set of lines that we have only genotypic information. So looking at the alleles that each line has for each marker, we can just add up all these effects and get an estimation of the breeding value of each line. This is an example for diastatic power, one of the traits for mold quality. And these are the estimated values for each line. These are standardized data, so we always get a value above or below the average of the whole population. These are some results of our prediction models. These are results of cross-validation using 3000 SNPs and rich regression. Again, this is the static power. This is in the X axis we have the genomic estimated breeding value. In the Y axis we have the actual value, what we call the true value of each line. And then we are building these regression lines. These are two different environments. For some environments, this prediction works really well. We are getting R-squares of about 70%. Other environments, it doesn't work as well, around 50%. And also for other traits involved in mold quality, the predictions are not as good as this. It depends very much on the heritability of the trait. So genomic selection is the way that we are using and we will be using to select double haploids in a breeding program for mold quality. But also there's several regions in the genome that we have a special interest in, and we still use traditional marker-assisted selection, not for making all our lines fixed for all these regions distributed across the genome, but at least we want to increase the frequency of favorable alleles at those specific regions. And we can select the parents for the crosses in a breeding cycle based on the alleles that they carry for these regions of interest. We can, once we have identified these regions, we can include these markers because for many of those we have the sequence SNPs linked to the QTLs responsible for these traits. We can design specific assays to genotype the parents and select or increase the frequency of these alleles in our population, or we can also include all these markers in our genotypic assays that we will use for genomic selection. But to identify all these regions, we still, oops, there's one missing a slide here. To identify these regions, we are still doing traditional marker-assisted selection and QTL mapping in mapping populations. We are doing association mapping to identify regions of interest. We are doing QTL mapping on biparental populations. This is an example of a QTL for cold tolerant, just recently found in our germplasm, and this is useful for our germplasm. So markers for this QTL are automatically added to our genotyping assay. And in general, as bringing everything together, we have used prior genetic information that we have collected for many years to identify regions of interest. We will include all the QTLs that we will find through the TCAP project. We will use double haploids for marker-assisted selection and genomic selection, and with this, we can rapidly respond to the challenges and opportunities of these shifting landscapes. And this is all. If you want more information about our program, just visit barleyworld.org and you will have lots of information about barley. Thank you, and I will pass this back to Pat Hasey.
Well, thank you all for attending our webinar this afternoon, and we're certainly open for questions. And if we actually do meet you physically, we'll be glad to share with you some barley foods or beverages, depending on your preference. Jamie is typing. Oh, thanks to all the presenters. Thank you, Jamie. How were the lines selected from the large diversity population for the flavor study? I'll tell you what, it's going to take just a second to answer these questions because I'm going to refer the question to the appropriate person. So I'm going to put Ryan onto this one.
We made selections from the World Core mainly on the PCA pot that we picked up. There are a lot of lines that were very similar. So first thing I did was to go through and eliminate all the similar ones, and then started to work from there. I had a slight preference towards land races. I thought that that'd be a good place to start for looking for new genes.
Right. Zerby here to answer Tyson's question. Yeah, so that is a very good question, and I think kind of the current answer to it is I'd say we have hopes to try and develop CSR indices for disease resistant susceptibility, and they will turn into plans once I get a chance to kind of look at the literature and on what people have tried to, because I've heard from people that they think that they can recall that someone has, or that they've heard of people trying to do that before. And so once I can kind of look the literature and see what there is there, and how feasible that may be, and then I will pounce on it. Oh yeah, so Jinli, so we so for NUE, so first of all, I haven't had a chance. I have all of the 3 main water use efficiency indices. I have all of those calculated for my plots for the water use efficiency trial. I haven't adjusted them for field effects yet. And I'm also planning on, we also took infrared thermometer measurements of the fields. And so I'll trying to figure out how I'm going to put all that data together. But for the NUE yet, I haven't finished, but I'll say since I only have data in the near infrared and red spectra, probably basically going to be using just the NDVI and red edge, and then the various the various wavelengths with it, that options people give for those. But yeah, that's the plans. Oh, and there are separate, sorry, Sarah, there are separate locations for the water efficiency and nitrogen efficiency. They are separated in both space and time. The nitrogen use efficiency trials were planted in the fall, and the water use efficiency trials were planted in the spring because if any of you have been to Western Oregon, you will know that. So we're pretty close to Portland. It's pretty wet here in the spring, but it actually luckily dries out quite well in the late spring and summer. So, and that is the advantage of having facultative lines. But shoot, it is the same entries, although it is the same facultative entries. The winter entries will definitely, we will definitely not be planting the winter entries for the spring
for the
ones.
group?
And we actually planted a few winter ones, and that was relatively useless.
Well, let's thank
But,
Pat, Araby, Ryan, and Alfonso for their presentation today. The next TCAP webinar is going to be on Wednesday, December 12th.
Oh,
and that will
right.
be Dr. Gustavo
And so
de los
actually, so the
Campos
water use efficiency trial
from the
will
University of
currently
Alabama,
is only being planned for Corvallis,
Birmingham,
but the nitrogen use efficiency
and
trial
his
was
webinar is
planted
going to
this
be
past year in
titled
Logan,
Factors
Utah,
Affecting
with David
Prediction
Hull,
Accuracy of
and
Genome Enabled
at,
Predictions,
in Minnesota.
Lessons Learned from Empirical
And
Studies and Implications for Breeding Programs.
Unfortunately, all the Minnesota
that's going to
ones
be a genomic selection
died during the winter
talk, I believe.
But we do have 1 year's data from David Hull that I have to go through, and we'll have then for next year we'll have data from, hopefully, cross my fingers, with Minnesota, hopefully both Minnesota and Utah.